For over a decade, scientists confirmed that an entire human species existed, not from a skull, but from a single child's finger bone found in a Siberian cave. They sequenced its genome and traced its descendants across Asia, but no one could tell you what it looked like. In 2025, that finally changed. Researchers put a face on the Denisovans, and what stared back may rewrite what it means to be human. In 2008, deep inside the limestone chambers of Denisova Cave, a team led by Peter Deravianko brushed away sediments to uncover a fragment of bone no bigger than a fingernail. The specimen, labeled Denisova 3, was logged, sealed and shipped from the Altai Mountains to Leipzig, where Svante Pääbo's laboratory began the painstaking process of ancient DNA extraction. What they found defied all expectations. The mitochondrial DNA sequence from this single finger bone did not match Neanderthal or modern human signatures. Instead, it charted a separate course, one that split from Neanderthals nearly 400,000 years ago. Within months, the nuclear genome confirmed the anomaly, a distinct branch of humanity, named not for a skull or skeleton, but for a genetic code alone. No other hominin had ever been defined this way. There was no skull to measure, no jaw to compare, no face to reconstruct. Only a sequence of nucleotides, a digital shadow in the archives of evolution. The Denisovans became a species known first as a genetic signal, not a visible presence. Their existence was undeniable, their identity invisible. The absence of a face made their story more unsettling, a lineage that left its mark in the bloodlines of Asia and Oceania, yet offered no features to recognize, no eyes to meet. For scientists, the discovery transformed the search for human origins. The fossil record had always been built on bones, but now a single child's finger was enough to redraw the human family tree. The question loomed, what did these people look like? And would the world ever see their face? A skull unearthed from the mudflats of Harbin, China, has forced scientists to reconsider what counts as evidence in human origins. The cranium, catalogued as IVPP V12345, surfaced not from a cave or a burial, but from the hands of bridge workers in 1933. For decades, it languished in museum storage, its significance overlooked. Only recently did a research team in Harbin subject the fossil to uranium thorium and electron spin resonance dating, revealing an age of at least 146,000 years. This places the skull in the same broad window as the earliest confirmed Denisovan DNA from the Altai, yet half a continent away. The skull itself is massive. Its cranial vault measures nearly 1,400 cubic centimeters, rivaling both Neanderthals and modern humans. The face is strikingly wide, about 140 millimeters across the cheekbones, with a broad nasal aperture and thick brow ridges that arch in a continuous line above the eyes. These features do not fit neatly into any known category. Some researchers point to the vault's shape and facial breadth as hints of a Denisovan affinity, suggesting the Harbin skull might finally offer an anatomical anchor for a lineage known only from genes. Others caution that the resemblance could be superficial, the product of convergent evolution rather than shared ancestry. Attempts to recover ancient DNA from the dense bone have failed, leaving the connection unresolved and the debate ongoing. The Harbin cranium stands as both a promise and a problem. It is the most complete archaic skull from East Asia, yet its true identity remains disputed. Without genetic confirmation, the face behind the fossil is still a hypothesis, an image caught between possibility and doubt. The question persists: can a face be reconstructed from a skull whose lineage is still in question? Or does the mystery deepen the moment we try to make the invisible visible? Every detail of the Denisovan face reconstruction rests on a pipeline designed to leave nothing to chance. Work begins in a sterile, clean room where ancient DNA is extracted from bone or tooth powder under strict contamination controls. Researchers wear full-body suits, double gloves, and face shields. Each sample is processed alongside extraction blanks to ensure that modern human DNA stays out. Only when contamination drops below 1% do they proceed. The extracted DNA is riddled with damage from tens of thousands of years in the ground. To correct for this, uracil DNA glycosylase, known as UDG, is applied. This enzyme removes the molecular scars left by cytosine deamination, a hallmark of ancient DNA decay, allowing authentic genetic signals to stand out from noise. Sequencing libraries are built from these purified fragments, then run through high-throughput Illumina platforms. The result is a digital archive of billions of short reads, each mapped against the human reference genome and filtered by quality thresholds. Once the genome is assembled, the next stage links genotype to phenotype. The face DNA database, built from modern genome-wide association studies, connects thousands of single nucleotide polymorphisms to specific craniofacial traits. Each Denisovan SNP is cross-referenced against this database. Algorithms predict the likely thickness of brow ridges, the width of the nasal aperture, the projection of the cheekbones, not as guesses, but as probabilities grounded in statistical models. These predicted traits become coordinates for a 3D morphable model called 3DMM. The basal face model, adapted for archaic inputs, serves as the scaffold. Trait vectors from the Denisovan genome deform the mesh, producing a digital skull that reflects both the Harbin fossil's geometry and the genetic blueprint of Denisova 3. Every step is logged, reproducible, and open to scrutiny. The face that will be revealed is not a work of imagination, but the end point of a process built to minimize uncertainty at every stage. A face, once lost to prehistory, now stares back, assembled not from imagination, but from the hard calculus of bone, gene, and probability. The Denisovan reconstruction, released in 2025, presents a visage both familiar and alien. Heavy brow ridges sweep across the forehead in a single, unbroken arch, thicker and more pronounced than those of modern humans. The mid-face is strikingly broad, with cheekbones that flare outward, echoing the massive zygomatic arches measured on the Harbin skull. The nasal aperture is wide and flat, set above a mouth framed by a robust jaw. These features are not artistic choices. Each was tagged with a confidence grade, a calibrated judgment distilled from dozens of genetic markers and fossil measurements. The supraorbital torus, rated high confidence, matches both the Harbin cranium and the genetic signals from Denisova 3. The breadth of the midface, too, earns a high grade, supported by both morphometric data and genome-wide association studies. Other traits, like the exact contour of the lips or the tilt of the eyes, are labeled with medium or low confidence, their details blurred by the limits of ancient DNA and the gaps in the fossil record. An annotated overlay displays these provenance tags: green for established fact, amber for working hypothesis, and red for preliminary guess. This system does not erase the uncertainty, it puts uncertainty on display, inviting scrutiny. The result is a face that feels deeply real, anchored in evidence, yet haunted by the shadows of what we cannot know. For the first time, the Denisovan is no longer a ghost in the genome. The abstract code has a brow, a nose, and eyes that once gazed out across Ice Age Asia. The mystery of their identity remains, but the barrier between past and present has thinned. The face is here, demanding to be recognized, even as it reminds us how much remains hidden. Denisovan traces stretch from the frozen valleys of Siberia to the limestone caves of Southeast Asia, leaving genetic fingerprints across a continent. Fossil DNA has surfaced in the Tibetan Plateau, the Baikal region, and as far south as Sulawesi and Taiwan, revealing a range that rivals any other ancient human. This reach is not just geographical but environmental. Denisovans survived in frigid steppe, dense forest, and at the thin air margins of the world. Their legacy is written into the bodies of living people. In Tibet, nearly 9 in 10 residents carry a version of the EPAS1 gene that allows them to thrive at high altitude, where oxygen is scarce. Genetic analysis shows this adaptation is Denisovan in origin, introduced through ancient interbreeding. The same gene is almost absent in lowland populations, a silent marker of Denisovan life in the Himalayas. Their face, reconstructed from bone and code, is tied not only to a vanished world, but to the physiology of people living today. A single genome can reveal more than 1,000 bones. In 2012, analysis of a femur fragment from Denisova Cave exposed a genetic hybrid, a girl with a Neanderthal mother and a Denisovan father. Her DNA, split almost evenly between 2 ancient lineages, proved that interbreeding was not a rare chance occurrence, but a recurring thread in our shared history. Yet the physical evidence remains stubbornly incomplete. No Denisovan skeleton has ever surfaced intact. Taphonomic bias, where bones are dissolved in acidic soils, shattered by freeze-thaw cycles, or lost to scavengers, erased most traces before scientists could ever search for them. The fossil record is a patchwork shaped by chance and chemistry as much as by time. What we know comes from fragments and genes, leaving entire chapters of Denisovan life written in code, not in bone. We know the Denisovans' DNA better than their bones. Their face is only now stepping from genetic code into flesh and shadow. Yet most of their story still waits in the dark. In every fragment, science reveals not certainty, but how much mystery our species still carries. What else remains unseen?